All right, class. Good morning. So I'm gonna create a tutorial for the lab, and then we're gonna talk about the assignments. So that assignment, that homework, will be due on Wednesday, 22nd. So either our lab is gonna be online, or our lab is gonna be in person. Regardless, if our lab online, then you will send it to me. And if our lab is in person, you will give it to me physically, because I have to have your first assignment grade, first quiz grade, right before the exam number one. Exam number one is going to be on Wednesday, the September 29th. And I will create bullets for the exam number one, so you would know exactly what to study for. All right, class. So I already sent you announcement about the Pearson study link, right? So let me go to students view, but I'm pretty sure that you already found that thing. So you will have this Pearson study area, right? You click on it, and there's a study area link. So you follow that link. That's this link is for everybody. So what I wanted to point out that if you are unable, or your browser is unable to launch this, it's just because of pop-up blocker. So I have the same thing, right? So what I have to do is pretty simple. So click here, and then this thing comes out, and then you reload the page again, you click send, and then it will come out, unless you can go into your browser settings and remove the pop-up blocker. So I think that most of you already figured that out, and some of you send me messages asking how to do that, and I think that you guys are already fine with that. So let me kill that. So that's our study area. So for our lab, so you go to the Physio X, right? Go to exercises, and the first exercise that we have to go through, that what we did in class, is a simulation dialysis, simple diffusion. So you can answer all these questions, go next, but that's not, well, I do suggest to read over, right? So read it. That's what we discuss in class. You can answer questions, not necessarily when you click submit, so I will not see what you're submitting. So next, submit or continue, right? So, and once you click submit or continue, you arrive to this thing. You have this, right? So you have that. So that's what you have to give it back to me. So all you have to do, just fill this thing. So the first assignment, there's going to be two assignments, right? So for the chapter number one that we went over for body orientation, so you have to give me back this. So answer these questions here, answer these questions, answer all of the questions except where the crosses are, right? So whatever I crossed out, you don't really have to answer that. So it will be in total like 15 questions. Then please answer this, and please answer that as well, including 8, 9, and 10. So do it to the best of your knowledge. If you do not know, do not stress. I'm gonna grade it very fairly. So because I do understand it's just the beginning of the human body orientation, so a lot of things are new for you. So even if you Google, because you're going to do this at home, please use it wisely. Use it as the educational tool. Just do not copy and paste everything. Use your own words, paraphrase, so you will learn it. All right? So that's the assignment number one. Assignment number two, so you should have that. So you have this all under course materials, under lecture slides, under lab number 2, membrane, right here, membrane transport Physio X worksheet. And the second lab is this, right? It's membrane transport. So membrane transport, please fill this, right, based on the task that you're gonna perform. Answer these questions. So the first one is diffusion, and the second one is osmosis, right? So it's pretty simple. Just log the data. What are we gonna get from here? I'm gonna do it. I will remind you how to do that. and then answer these questions to the best of your knowledge. Again, if you are struggling, do not stress it. I completely understand this is all new to you, but again, if you copy from somewhere, use your own words, paraphrase it, so you will learn that way. So let's say we want to do a diffusion from one compartment to another compartment, right? So we have to put a membrane, right? So we put a membrane, and this membrane has a molecular weight, right? So cutoff. So molecular weight cutoff. So basically what it is this is the membrane, and the membrane has its channels through which, or pores through which some molecules can diffuse. So let's say, right, so let's say they're small, right, or they're big, or they're bigger, or they're really big, right? It depends on the membrane, it depends on the compartment. So if the molecule is this big, obviously it cannot go through. If this molecule is this big, it can pass, right? So let's say this is 20, this is 50, this is 100, and this is 200. So it is very easy for molecules to penetrate. So that's a diffusion. So it goes from highest concentration gradient. So what's the highest concentration gradient? So let's say on this side, right, on this side, there are a lot of molecules. And on this side, there are no molecules or a few molecules, right? So here on this side, there is the highest concentration gradient of this particular solute. And on this side, there is a low concentration gradient. So through the law of diffusion, molecules can diffuse from high concentration to low concentration gradient. So, but it's not enough, right? Because it depends on the pore size, right? Molecular weight cutoff. If, for example, right, so they diffuse with their own, let's say, kinetic energy. They have kinetic energy because molecules, they bounce one from another, or molecules always bounce off of the walls of the compartment, and they bounce back. So, but if the pressure would be created, some sort of a pressure, right, or the piston, for example, you put a piston here and you create a pressure, and that piston is being pushed down, so it creates a pressure, so there's less space, less volume, so the molecules will collide over the walls or over themselves a lot faster. So there's going to be many collisions. So then if there are many collisions, so there's more pressure, there's more kinetic energy. So maybe it is possible to squeeze one molecule, like bigger molecule, through a smaller hole. But it would be a lot easier for this molecule to penetrate through this membrane. And when we're conducting our lab test, right, so this, what those membranes are for, so we can see how it's all done. Right, and we record what time it takes, right? How long it takes for these molecules to diffuse. So this is a diffusion. Obviously, for the sodium that we're gonna perform, right? Right. So 20, it's not gonna be able to diffuse because I guess it's too big. And then it's gonna diffuse through 50, 100, and 200, right? So if it diffuses through 50, then you don't even have to collect extra data for 100 or for 200. The reason we're doing this for 100 or for 200, we want to see if the time frame will change. So if for, let's say, 50, it will take 10 minutes, and for 100, it also takes 10 minutes. We know that this molecule is small enough to diffuse with the same amount of time. That's it. If the pressure would be created, right, it would take a lot less time. And through this big pore, right, maybe a lot of molecules can go through at once, right? Instead of one molecule, single file, right, if it's a bigger hole, maybe a few molecules can penetrate. Right, let's take 20 membrane, right, and we go for 9 millimoles. So we'll go for 9 millimoles, we dispense this, then we deionize water. Deionize water, meaning it means that there are no particles, there are no ions, right? So if this is a compartment and there are no ions, right? So there are no sodium, chloride, potassium, there are no minerals, let's say. So it's like distilled water, right? Distilled water, there are no minerals in it. So that's why we call this ionized water. So then we click start, and it takes time, right? So this was like 60 minutes, 1 hour task, but obviously it's going to go faster. So I think it takes 10 seconds. I don't know why we need to wait this 10 seconds, but so no diffusion. So what it means is that this molecule, right? So this molecule is too big for this hole in the membrane. So molecular weight cutoff for 20 is probably small enough. What if we go with the smaller amount? Let's put 1, right? I just want to see what's going to happen. Dispense deionized water. Dispense, because when you're creating, when you're working on something, when you're conducting a test, you would want to test all of the possible possibilities. So to eliminate bias as much as possible. So no diffusion. So it's not just the amount of particles, it's the size. We know that, right? So we take same name 9, right? Let's go for 50. We'll go for 50, we click start, we wait. So what I'm doing right now, I'm just using this one screen, but this particular simulation will ask you to go next, next, next. So equilibrium takes 10 minutes. So we know that the hole here is big enough. So then I can go already directly to 200, click start. So 10 minutes, same thing. All right, so, and you do this for all of these molecules. You can actually, because the first test is asking to use 9 millimoles, right? So use 9 millimoles for 1, then you use 9 millimoles. Also here for urea, and you again can start with 20, right? But with 20, I'm gonna tell you the answer. The 20 is nothing, but I want you to perform this test so you can see. So let me use 200 right away. So there's a different data for each solid. So deionized water. So for sodium, it's still obviously those 10 minutes because we already did the test. For the urea, it takes 16 minutes. For albumin, there's no diffusion because albumin is too big for that. And for the glucose, it's 36. So what happened if this is your membrane? So 20, 50, 100, and 200, right? So sodium, 10 minutes. Through 200, right? So through 200. For the urea, it took 16 minutes, right? Also, for glucose, it took 36 minutes, right? And for albumin, it took 0 minutes. Why? So why do we get these results, right? 36 and 16. So it's just because of the size of a molecule, molecular weight. So let's look at the sodium. So the sodium's molecular weight, I didn't calculate it. For urea molecular weight, 60.6 grams per mole. For glucose, 180. I will round this up. And for albumin made from 167 amino acids. 167 amino acids, and the weight of each amino acid approximately 135. So 1 amino acid, that's the size. So multiply by 600 amino acids. So you'll see how heavy, big this molecule is. It's not just the weight, it's the size, right? The more weight, so the bigger the size is. So if you remember from chemistry class, so that's how you calculate molecular weight, right? So you get the molecule and you reference to periodic table for the molecular weight. So I do recommend to run it one by one, so you will have data because you have to put the numbers right here. That's for the diffusion. So the next one is osmosis. So osmosis is a little bit different. So for the sodium, it is 8 millimoles. For albumin, it's 9, and for glucose, it's 10. So I do suggest to run it one by one. So osmosis versus diffusion, it is another process where when there's more molecules on one side and there are less molecules on another side. So by molecules, I mean by solute, right? Solute. So, and let's say equal amount of water here and there, right? So that's water, equal amount of water on both sides. So in the process of osmosis, it is the movement of water from low concentration gradient to high concentration gradient, because water will be attracted by the solute. So for our test, right, again, so we're going from left, we're going to right. So there's nothing, so it's a deionized water, there's empty, here nothing, and on this side we have solute, right? So it's the movement of solute from one area to another area. So we're moving from this to that. If we place distilled water on both sides of the membrane, net movement of water does not occur. Remember, however, that the water molecules would still move between the two sides of the membrane. In such a situation, we would say that there's no net osmosis, because osmosis would be pull over water. So that's why in our case, in our test, when we are performing the test, this is the water without minerals, deionized, minerals been removed, molecules been removed. The difference between distilled water and deionized water is that they called it here is distilled, meaning that there are no solutes. But if I'm not mistaken, distilled water, minerals are removed, ions are removed, but there's still hydrogen and hydroxide. Do they really have osmotic pool? So actually it's interesting, guys. Look it up if hydrogen or hydroxide has an osmotic pool similar to sodium. So also I want to point something out, that solutes move down its concentration gradient. So the solutes move from high concentration gradient to low concentration gradient. Water also moves down its concentration gradient from high to low. But in osmosis, the solute has their pull, so the water will go towards highest concentration gradient of the solutes. So you can perform and record the data for all of this. Please do it one by one, so you will have the data in each square, and then you answer these questions. So, and the due date would be September 22. I will create an announcement on that, so it would be a reminder for you. All right, so please do those two labs, one for body orientation and one for the membrane transport. I see you in 2 weeks.